everybody. Before starting this masterclass, we would like to ask you a question, simple question. Do you know the difference between a reactive approach and a proactive approach to healthcare, and the advantages of a proactive approach? In a few words, somebody can answer this question or not? Nobody?
No, nobody.
So we have to present this masterclass.
Yes. Let's go to the answer.
So, as you know, even today, healthcare systems are often focused on treating diseases and only reacting to symptoms once they appear. But I suppose you know these systems are no longer adapted to the current needs. For example, an aging population, but also chronic diseases and social issues such obesity, addictions, and sedentary lifestyles, of course. And so there is a real need for a transformation today adapted to the demographic, but also the societal and technological challenges of our 21st century. And with the rapid evolution of technologies, of innovative technologies such as AI, of course, and a growing need to optimize resources, we must take a proactive approach. And this transition to personalized care, supported by innovative technologies, so I cited AI, of course, is essential to improve health, public health, in a, I would say, a holistic and also an effective manner. And even today, and traditionally, medicine has been reactive, focusing on diagnosis, focusing on treating diseases, illnesses, once they appear. And today, it is becoming increasingly clear that to improve public health, we must take a proactive approach. That means simply anticipating health problems before they appear, and not just reacting to symptoms once they are here, as I said previously. So, but proactive care is a long-term approach that involves, of course, regular checkups and lifestyle changes to prevent, of course, future illnesses. So, as you can see here, 5P medicine, that means preventive, personalized, predictive, participatory, but also event-based. So event-based, it's proof, represents a fundamental transformation of this proactive approach. And that means we treat each individual as a unique case, taking into account several characteristics, such as genetic characteristics, and also lifestyles and environment. And the integration of innovative technologies, so AI, of course, but also data analytics, plays a crucial role in this transformation. So I mentioned AI, of course, because we are at the Adopt AI International Summit, and AI offers unprecedented opportunities to collect, to treat, to analyze massive amounts of data in real time. And for example, if I can give you some, using predictive algorithms, we can identify individuals at risk of developing diseases, but also enabling early and targeted interventions. We have the same aim with other tools, such as mobile health tracking apps or wearable devices. And this approach, of course, helps optimize healthcare systems resources, and by investing in prevention rather than treating advanced illnesses, we can ease the burden on hospitals and improve the overall efficiency of care, of course. And collaborative care models, patient-centered care models, also incorporate technology, as we know, and promote the exchange of information between the stakeholders involved, of course, the healthcare professionals and the patients. We talk about 5P medicine, we talk about innovative technologies, and 5P medicine, enhanced by AI now, is not just a future vision. It's already being implemented in different healthcare systems around the world, and the pilot projects are showing promising results in terms of improving health outcomes, but also reducing costs. So I propose you to have a look at all of this with a concrete example with Alexandre Bouthillier from Linearis, precise from Canada.
Thank you, Evelyne. So I'm going to drive you to the short story of AI, and please remember that this 5P medicine are kind of the 5 fingers of the hand. They're very tight together, but the first one is really the proof. The proof is in the data. So we need to start with the data, and essentially AI can have a look at those data. And we started in about 2012 with a model called AlexNet, and it was not because of me that the model was named AlexNet, but the modern AI was co-invented in Canada by the group of Yoshua Bengio, Jeff Hinton, and in New York, but this is close to Canada, it's soon be part of Canada, Yann Lequin in New York. So AlexNet really make the breakthroughs in images. As you know, radiologists, now most of the FDA, most of the CE mark in Health Canada, a medical device approved as a software are in the radiology. But as you move up from those perception AI to the generative AI, and I'm sure everybody's using ChatGPT. Who's using ChatGPT? Please don't be shy. Okay. Who's not using ChatGPT? Typically, there's one person in the. Okay, there's one person. 2. Okay, we need to talk. But for all the others, what GPT stands for? Somebody knows what GPT stands for? Okay, there's one nerd like me in the room that knows what GPT stands for. So I hope you'll learn at least one thing today. G for generative, so that was an easy one. It generates a new token that will minimize the error rate. So when you read a sentence, of course, you want the full sentence to be readable and with no errors. P, and this is the important concept, is for pre-train. And T is for transformer, the technology that still dates back from 2016-17 in a paper called Attention All You Need. So let me go back to the pre-train. Pre-train was trained on all available data, publicly available data, not supposed to be on private data, that are textual. We recently started to incorporate images, some videos, some audio, but that's about it. The physical world, this is where we're embarking, is much more than text and images. I can guarantee you that. And we just had an example before this talk on ECG, EEG, other type of data. We can talk about Raman spectroscopy, ultrasound. So there's a broad breadth of data that sits in the physical world that is currently not part of those large language models. So imagine for a second that we incorporate like probably 10, 20 times more data in those models. Maybe we'll get a better understanding of the real world. And I had the opportunity to meet with Jensen Huang. Jensen is the CEO of NVIDIA, one of the largest company in the world, almost reaching 5 trillion for a quick blip. They've now launched a computer that sits on your hand that has almost a petaflop. That's the size of like a modern supercomputer. So you're able to launch a local ChatGPT-like on the palm of your hand, like about a little bit bigger than this, where you can run those models with a reasoning model, with models that are specific to your domain, but also experimental data to better learn and to better understand the proof, the first fundamental element of the 5P. And this is very important because people are starting to do that more and more, getting their local LLM, and we call them RAG, or now there's a new technology called MCP that really connects to your local database, but you can do that really locally. So I'm going to go into a real and concrete example on a data that I hope you'll understand better at the end of this talk. So you learn about the acronym of GPT. But let me go into where we're heading. So I told you we use AI to save time, to augment physician capability, mainly in the radiological space. So this was good, but this was not transformative. Where AI can really transform is by predicting. We want to predict what will happen based on the proof, based on the data, if we do something or if we don't do something. So the transformation part is where we are, and where we hope to go is to be able to predict the response, predict toxicity of new treatment, to look really on actionable insights. So let's talk about very important data, the omics data. So if I ask outside of this room, people will know about genomics. If I ask outside of this room about proteomics or proteins, people will know, oh yes, there was this AlphaFold where we can fold a protein like for real. But if I ask people what are metabolites, maybe I can ask that question in that room, and not everybody knows about metabolites. So who knows about metabolites? Okay, so who knows about cholesterol? Cholesterol is a metabolite. Vitamins, hormone, amino acids. So I won't name them all because there's 250,000 metabolites that really regulates our body, transform what we eat in energy and matter and control the waste. So why don't we measure more metabolites? I mentioned the amino acids, which are very important when cancer is lurking on. We do this actually in the newborn. So actually, to be clear, the 220,000 entry in the HMDB, the Human Metabolome Database, which is hosted in Canada, we have world expertise in Canada. We are able to measure on a consistent fashion almost 1,800 in something that we call the GigaKit. So it's not like one marker. 1,800 metabolite that we measure in blood or feces to really understand all type of disease and to better understand the metabolomic disease signature. So this is important to understand that this is not a new type of data, but it's an expanded type of data. Why I say that is because we've been doing metabolomic testing in newborn screening, in the newborn test, since the 1970, if I remember correctly, 1972. In 80 countries, we do that within 72 hours that your child is born. And it identifies between 25 and 50 diseases. It depends on the country. Those are done for genomic disease, even though that we measure the metabolomic impact. So the question I have for you is, if you're lucky enough and you do an annual checkup, why don't you do a large panel? This is not done in adult. We do like maybe 10, 15 things, but not something as important as for the newborn. So I'm very happy that we do it for the newborn, but we should use that technology more in adults, in elderly, to really understand the disease and what does it mean quantitatively to have that disease. So almost 1,800 marker in 24 minutes with 4 or 5 drops of plasma or serum. So this is done in mass spectrometer. And let me go back to a disease that you all know, COVID-19, which is a protein disease. We wanted to test if COVID will show in metabolites. So we've tested the first graph on the top left in orange. Those are 6,000 proteins and metabolites that were measured. And we ranked them from the least significant, the least unbalanced, to the most unbalanced on the top left. And in blue, if we zoom on those, this is the graph that you see on your right, there are 17 metabolites that are unbalanced when you have COVID. Not 1, not 2, 17. And this is typically the error when we want to either diagnose a disease or specifically develop a new treatment for a disease or monitor a disease. We focus on the main pathway. But diseases are a little bit more complex. So this is here, 17 marker. if you're developing a treatment, should it work only on the first one? No, it should work on all of them. But if you're developing a new treatment and you test it, and it only works on the first one and it reduces it to 50%, that's information. That's information that you use, that you can refine your treatment to better make new medicine in that 5P. And what is missing and what we're working on is this concept of lab in a loop. We really want to provide to the next generation of drug discovery those metabolomic signatures. So this is the signature of COVID, but cancer will have a disease signature, neurodegenerative disease will have their cancer signature, infectious disease will also have their disease signature. So we want to provide those pathways to the generative model, not only the physical property, the chemical property, but really the pathways, because they're documented, we understand them, and we want to teach the AI, if you develop a new treatment, this should have an influence on those. So then when we go in the testing phase, please try to use more than something that is a mouse model. We've realized 2 years ago that humans are not mouse anymore. This was a big announcement from the FDA. They say you can go in clinical trial without using mouse model, but you still have to show non-toxicity. So, of course, the mouse won't disappear overnight, but using organ-on-a-chip, organoids, human cell line is a way to go. You can test those new drugs using those organ-on-a-chip in the same way and see on those 1,800 metabolomic markers what's the impact quantitatively for each of those markers. So this is an example of an organ-on-a-chip, very tiny. We're able to see the vasculature, the permeability, measure the toxicity and the response of a new therapy in human cell line, either hepatic, renal, or epithelial at high throughput. So this is very important. AI cannot go fast, where you can invent 1,000 new molecules per hour, and then you are able to test once a week with mouse model. You have to make sure that what you invent in silico is matched with the physical speed. So this is for treatment. We'll go back on prevention, but you can understand that if we have that disease signature, we can do better prevention, monitor the action on prevention, and on early diagnostics. So to that end, we're establishing collaboration with the Sorbonne. We're establishing collaboration with Japan and Osaka to really expand this large metabolomic profile and to make it a de facto standard. We call it one test for all because it's the same panel that we do whatever the disease. It covers already 34 families. We think it's large enough to better understand the disease that will affect our loved one. Finally, you know the drug industry is very long, is painful, and AI can have a lot of application, not only in the drug discovery phase, but in the logistics, in testing, and specifically there was a talk yesterday by Sanofi where the CEO of Sanofi said, please, can we go from 90% failure of the new drugs that we invent to 80%? I said, Paul, this is a tall order, like you want a 10% reduction, but this is what we could do. We could kill new drug faster, spend more time on the more promising one, and spend less time or refining the one that will show less response and less toxicity to actually reduce that 10 plus time to maybe 6-7 year time. I'll end on this slide. I hope you understand the importance of getting a full omics signature. I think we spend too much time on the genomics. It's important to understand the genomics, but the genomics is only your book of life. It's not because you have a predisposition of having high cholesterol that your cholesterol is high. But if you measure your cholesterol and your cholesterol is high, guess what? You have high cholesterol. So for simple disease, there's a one-to-one match with metabolomic measurement. For more complex disease, you need a signature. And you need to feed that signature in the prevention, in early diagnostic, in the right treatment, personalized, because not everybody has the same metabolite, to monitor the disease state, how does disease evolve, to make sure that you can predict the next action, and least but not least, the discovery. How can you use that disease signature, and I just showed you how, to be able to measure the health impact of each of those 4 segments. This is a $200 billion market. To go back to the drawing board and see how we can refine our understanding of the disease, because we don't think we understand them very well, otherwise we would have treated them all. So that quantitative feedback loop is very important.
Thank you, Alexandre, for the great presentation and intervention, of course. And to conclude this masterclass, very short masterclass, we apologize for that, of course. Moving from a reactive approach to a proactive approach, which is the topic of this masterclass, is not only a necessity, it's really an opportunity. And by the integration of 5P medicine and innovative technologies, as we saw, so AI, for example, we have the opportunity to completely transform care pathways, making personalized healthcare accessible for all. And this could be, well, the key for improving public health, but also optimizing resources and ensuring a future where each of us, all of us, will have the right tools we need to take control of our health. And to finish, by adopting this new vision, we will take a decisive step towards a more, I would say, inclusive and efficient healthcare. So thank you for your attention.